right, thanks, Adam. And this is a beautiful place I get to work, Yellow Bay there. It's the eastern shore of Flathead Lake looking north towards Glacier Park. And that's the Flathead Lake Biological Station. The title was already shown. It's a paper that Adam Sepulveda and multiple other authors have in Review It, Trends in Ecology and Evolution. And it also has a couple take-home messages that are in my talk that eDNA can work as a tool for management, but there is a need for improvement, especially in communication with managers and interpreting of the data. I just wanted to mention that the data I'll show are produced in a lab overseen by Steve Amish, so I wanted to acknowledge his help, and Phil Matson, who organizes most of the sampling, and many others who have helped. So first point, eDNA is a mature science and reliable monitoring tool, or can be. There's a dedicated journal that was initiated last spring by Wiley called Environmental DNA. There's a book that was out in 2018 by Taberlet and co-authors called Environmental DNA, and many review papers going back at least 5 or 6 years. One of the quotes from this paper that's helpful to us is, the strength of evidence depends on the frequency and consistency of positive detections in time and space. So one detection of DNA in a sample is not really useful. It needs to be repeated. And then some of the many review papers are listed below, and from them comes this conclusion that evidence is now extensive that eDNA detections can be reliable and often precede visual detection of invasive species. So we've been, since 2012 and a little bit earlier, sampling lakes around northwest Montana, Flathead Lake's in the middle there, and mainly for zebra mussels and quagga mussels and Eurasian milfoil, but other species now, from at least these 32 lakes designated with stars. And I just wanted to acknowledge and thank collaborators from over 10 agencies for helping sample and send samples to the lab, especially the U.S. Forest Service folks up in the Swan and in the Flathead region who've helped with the development and application of these tools, and the tribes, Salish and Kootenai tribes, who help a lot with sampling on the southern half of Flathead Lake and other areas. So we've been working to increase probability of detection, so sensitivity of these tools. And so one of our research questions has been, do large volume samples from a plankton tow net, like seen here on the end of a pole, provide earlier detection of aquatic invasive species than widely used standard filtering methods? And so you can drag this net in the boat, around a boat, if you want sort of a point estimate, or we use it on a lot of docks and piers. And so you would drag this, if you drag the net about 100 meters, you get over 7,000 liters sampled. And you wash the material down like Delaney's doing there with the spray bottle. She's washing the material to the bottom of this tow net. And then that material is taken from the bottom of the tow net and put into a tube filled with ethanol to make sure there's 80% ethanol in there to preserve the DNA, and that's sent to the lab. And we have sampling demonstration videos if you're interested. And so we've compared this to the more widely, very widely used filter method where whereas the tow net is a 64 micron pore or opening, about the size of a human hair, so it won't clog and it's very large. These filter samples, you can see the arrow points to a filter in a cup. You stick the cup in the water, and then this peristaltic pump pulls the water across the filter, a little disc, a little white filter disc, and the pore size typically is very small, 1.5 microns, maybe up to 3 microns is sometimes used. And the problem with it is it clogs after often less than a liter. So as the standard widely used approach, we've compared those two sampling approaches at multiple locations. And I'll show you some data. And the take home is almost always get far more DNA from a tow net. And so there's a publication, Sepulveda et al., that just came out showing this is true for Montana streams for bull trout. Schabacher et al. Is in review, and that has data showing the same pattern with more DNA detected, more positives for the tow net. For milfoil, native and invasive milfoil, as well as mollusks, and then another paper I'll show some data on that's just in the preliminary stages of development. So after getting data from multiple species in Montana, we wanted to see and test zebra mussels. So we went to Minnesota, and we found that tow nets detect more zebra mussel DNA than traditional filters. And so we found, with the help of Mike McCartney out in Minnesota, a lake that was just recently invaded. And this map in the middle shows North Star Lake among the more than 10,000 lakes in Minnesota pictured on the left. North Star Lake was invaded in 2017. The red dots in the middle eastern or right shoreline were detections from plankton tow nets where they detected veligers by microscopy in 2017, and divers then came in and also visually detected some mussels in those areas. So in 2018, we went there, and we sampled at these two locations. Number 2 is relatively close to the detection points, and one's farther away, so we predicted that we'd probably get more DNA in the sampling location number 2. And we took 5 independent tow net samples at each site and 5 independent filter samples at each site. And I'll show you a table with DNA copy number results. So I showed you the sample. We extracted it in the lab, and we used a quantitative PCR assay published in Gingera et al. 2017 and another assay. And we just take an aliquot, or a small volume from the extract of each sample from a tow net and a filter. And the top right shows zeros for the filter at site number 1. And the tow net sample had from 51 to over 400 DNA copies. And all of this is repeated, so these are averages over at least 6 samples. And then site 2 did have more DNA. You can see 4,000 to 10,000 DNA copies detected. And 3 of the samples were above the limit of detection, which we call right at about 10 copies. We wouldn't call it a detection at that site further north. So we've done this at Lake Winnipeg. Has a lot more mussels since 2013, actually, the first detection. And the tow net at these 13 sites was compared to filters at the 13 sites. And the tow nets detected up to 500,000 copies, 50,000 to 500,000. And the filter samples from 0 to around 5,000 copies. This summer, we went to Lake Bemidji, unfortunately a nice lake in Minnesota. It was also invaded in the last year, and we have similar results as well. It's a very new invasion, so there were some zeros even with the tow net, but all zeros with the filter, and up to several hundred DNA copies detected with tow nets at that lake. So in August then, Phil and others went to Tiber with help and coordination through FWP and others, and took 42 tow net samples at approximately 40 sites, and there were no dracenids detected. I'll just mention that the first and main assay we use is genus specific. We say it will detect either the zebra or the quagga mussel. No detections there. And at most of these sites, there was a deep tow net near the bottom, 3 to 5 feet off the bottom, and one near the surface. So that's good news. So conclusions, I would say we need more sampling in time and space, and we could be cautiously optimistic that there's either a low-density population, maybe a declining population, or we hope it might be extirpated. But absence of detection of something doesn't mean it's not there. So we'd like to go back and sample at more locations. I'm going to mention a new technology developed by Cody Youngbull in our group at the biostation. It's a continuous flow digital PCR machine, so samples can continuously be run through the machine, and about every 20 minutes, 20 to 30 minutes, you'll get data on 20,000 independent PCR reactions at very small volume. And so it allows sort of real-time tracking or localization of, maybe through triangulation or looking at gradients in DNA quantity across the lake. So Cody took it to Lake Mead. There's 7 circles here where they sampled in a speedboat at these sites one day. And what they found indeed was a gradient or more DNA copies near known colonies near shore. So 1 in 7, for example, had more DNA copies, 9 to 20 as opposed to 3 to 6. So this technology has potential. We did use it at Tiber with no detections there either. And we're further working on it with Cody to put known samples in a location and then sample at different distances to see if we get a gradient effect. So conclusions, the high volume sample TONet approach almost always detects more DNA than filters in lakes and streams for multiple species, from milfoil and multiple mollusks to mussels and bull trout. So researchers and managers should consider using large volume TONet samples or other large volume approaches, because while the amount of DNA per liter is actually less than a filter sample, since you have thousands of liters, you end up with more total DNA. And we do get more total DNA, which can be an inhibitor if there's so much DNA in there, for example, you have to dilute the sample. Or we haven't had problems with inhibition. And coverage is an issue because if eDNA is patchily distributed in space, to have a high probability of detecting the patch, you would need many small samples distributed, which becomes expensive, whereas a TONet more often would pass through the patch, for example, and capture the sample. And one last point, we don't know, but I suspect the pores are getting clogged, and so the effective size of the pores are less than 64 micron. They might be really small towards the end of your toe, so there's more work to be done. And the real-time tracker has potential to localize invasive colonies. I was just going to mention how we might help bridge the gap between research and application by managers. And it will help to develop a plan in common between the lab and the managers that integrates uncertainty in eDNA results into the decision making. And so, for example, as was mentioned, a positive hit does not mean there's alive organisms. And you need multiple positives in time and space, in my opinion, to have much certainty that there's likely an organism there. And then, as in the paper suggests, and Adam mentioned, and I believe a lot of you have access to a decision tree draft, use a decision tree with best practices for both the field and the lab that integrates trend of positives over time, and that includes your risk tolerance for false positives versus false negatives. You could establish an MOU with an agency as to how the results will be reported or communicated. That can help build confidence. So the last thing I wanted to do was just briefly show a video of a new technology that's just outside that door over there, that we have put a TONet behind, and we can send it down, for example, to the thermocline. If a lake has a thermocline, they still should exist in many Montana lakes right now. Because it's been seen in Lake Mead and other places that villagers may collect, or DNA and cells may collect on the thermocline, or you could take the net down near the bottom. And Phil, you were going to talk about it briefly?
First of all, I would thank you, Adam, for allowing me to weasel my way up here. Thank you to Western Regional Panel for allowing me to weasel myself onto a table in the back corner there with my colleague, Kyron Zimmerman. I want to show off an underwater rover that we have attached a plankton TONet to in the hopes of sampling better. As we went to Tiber Reservoir, our idea was to take a surface tow using these plankton TONets, and then also target the bottom or go through the thermocline. As Gordon mentioned, a lot of that DNA or the veligers might settle down to the bottom, so that's a really key area that we like to target. However, I was very disappointed that I did not find DNA at Tiber Reservoir. Not that I want to see it, but I want to be the guy who finds it, because if it's out there, we want to find it. I'm not just sampling to cross it off my list, say that I sampled and be on my way to do another job. We obviously all care about preventing the spread of these mussels, so we're trying the best that we can. So the goal of using this underwater rover, first of all, was just to see if it had enough power to pull a plankton tow net. Those tow nets get filled with debris, they clog up, they cause a lot of drag. And here we go. Nice live video feed. So with the plankton tow going down to the bottom, we can't always ensure that we're drawing a straight line down to the bottom. Sometimes the currents will bring our net up. We want to be careful that we're not getting too close to the bottom, because then we'll get clogged up with a lot of sediments. And then we also wanted to see if we could find the thermocline. So boom, there we are. We hit the bottom. We've got some nice lights, though, to this thing, so we can clearly see the bottom. But right there is the thermocline. You see that little shimmering right there? So with this underwater rover, we can visually see the thermocline, in addition to the on-screen video display that shows temperature and it shows depth. So we could see how far down we're going, and we could see the temperature. We can make sure that we're seeing that thermocline for sure. Kyron Zimmerman is the gentleman who owns this machine. We're partnering up to try to sample the best we can, and we're looking for partners to get behind us. So with that, thank you very much. If you have questions, you could see us privately afterwards. Thanks, Adam.
Anyone has questions? What's the difference in your cleaning protocols for the nets if you're looking for eDNA versus intelligence? Between sites, so you don't get a...
Right. So what's the difference in the protocol for cleaning the nets if you're going for eDNA in addition to or instead of villagers? We need about a 20-minute soak in high concentration bleach with some agitation. And so we often will then put a 30 to 40% bleach solution in a tub and put the net in the tub. And it will be there when we move between sites. The best is to have all the nets for the day ready so you don't need to do the decontamination until the end. We also have, to spray material off the net, a backpack sprayer we just started using, where after we take a sample, we thoroughly clean it with distilled water. Does that answer the question?
Something that we've been doing, though, is we've partnered up with Fish, Wildlife, and Parks to collect villager samples and eDNA samples. So we'll take 2,000 gallons of water, filter through our nets, and we will split that sample. One half of that goes to the Helena AIS lab, and the other half goes, that was for villagers, and then the other half goes to the Montana Conservation Genetics Lab at the university looking for eDNA. Now, when we decontaminate using that protocol, we first
Yeah. Yeah, it's a good point. If the net clogs and you want to be quantitative and know the volume that passed through the net, you wouldn't want to run the net so much that it would clog. And you could put a flow meter in the net to know when it stops. We're trying that now when the flow rate declines. On the other side, if you aren't so worried about being quantitative, you want to run that net as far as you can to cover as much water as possible, and a little backwash may not be a problem if you're not going to quantitatively estimate the volume you sampled. If you would just want to detect critters.
It's true, but I mean, at some point in time, they actually stop. They stop filtering water.
But it's a good point in question in part because a lot of tow net sampling is being done, and with washing of the nets or a separate net, you could, in addition to veligers, have an aliquot sent that's not being used anyway for veliger microscopy sent for eDNA testing.
Thanks, Gordon and Phil. I think it's also important to point out Yale with Bureau of Reclamation. I mean, that's another example of a group that's been using this plankton tow approach to not only look for veligers, but also eDNA. There's some differences between the two approaches, and that's something that we hope to, probe a bit more about, what are some of the potential repeatability, reproducibility, sensitivity trade-offs between an approach like Flathead Lake is doing versus a similar yet very different approach that Bureau of Reclamation is doing. So these are all things that merit further research to make sure that we are able to provide you guys with the best science in these tools, and I think most appropriately, best uses and limitations.